The Phoenix Friends of the Arizona Cancer Center have for 26 years supported our efforts in cancer research. Because of them, we've done some amazing things. One of our most recent examples is Dr. Marty Pagel's work. They gave Dr. Pagel $10,000 2 years ago and $25,000 this past year that have turned into over $5 million of grants, where we're going to be able to image tumors a day after they've been given a drug rather than 6 to 8 weeks.
So right now, if a patient comes to a relatively routine cancer clinic, there's a menu of drugs that can be assigned, and while there can be some very general tests done, even some DNA tests these days to look at the type of tumor, they're really only going to, there's a very simple process of choosing which kind of medication is going to be given to that patient. But if we can get a variety of information about the acid content, other molecular compositions as well, then we can start to really prescribe the type of medication, the frequency of medication, the dose of the medication that's really specific for that patient. We can finally do personalized medicine. Should we run those in triplicate? A very good example of that is some research that has been funded by the Phoenix Friends, where we study the amount of acid within a tumor. Just like when we exercise and build up lactic acid in our muscles, we feel the burn. Tumors are also very metabolically active and producing a lot of acid. Well, that has a major consequence for treatment, because a lot of our common treatments are then resistant to those acidic tumors. The problem is we don't know which tumors are acidic and which ones are more neutral in their acidity. There isn't a good technique to diagnose that. Right now we just use a wire and poke into the tumor and see what's going on inside. So I have a method now that we've created with magnetic resonance imaging, where we can tell the amount of acid within the tumors. Now we can tell a patient, don't take this kind of drug because you have an acidic tumor. Instead, take a different type of drug, even before they started that medication. Now we're working with Siemens Medical Solutions in Germany and North Carolina, in the Pittsburgh area as well. And then also in Phoenix. We're actually going to start our first clinical feasibility study in the Phoenix area. Barrow Neurological Institute up in Phoenix is also very interested in our techniques for looking at the acidity within glioma. And these are in human patients. We're not talking about just small animal models anymore. A lot of the researchers, including myself, have had family members and friends affected by cancer, and so we take this very personally. It's a war on cancer, and we like to say that, yeah, you could take a day off now and then, you could take a vacation, but cancer doesn't take a day off and it doesn't go on vacation, and so we're really driven by that. There's a lot of perseverance and enthusiasm in our cancer center to continue this research for that reason.
The Phoenix Friends have also supported very basic science research. For instance, the way they've helped Dr. Ann Kress. Her work is not only personally fulfilling, but she's trained the next generation of scientists. Many of the folks in her labs who've benefited from her work are now the directors and lead people at other cancer centers around the nation.
The focus of my laboratory is to really understand the molecular mechanisms of cells when they migrate and metastasize to other parts of the body. So what we're trying to do is understand the essential features of that so that we can block it and prevent it from occurring. Because now we know that in cancers, many cancers, there is early detection, but we really need to prevent the disease from spreading. And one of our most interesting discoveries very recently has been that that cells move in collections. They move as collectives, not as individual cells. And this was quite a surprise to us. You can mimic this in tissue culture, and we've optimized that so that now we can see when you stimulate these cells to move, they actually grow out, and they'll grow out at you. In a tube. So we sort of call this tube travel. And these tubules that form are also found in human tissue. So we've gone back to our pathology colleagues who are one floor down from us and confirmed that this does in fact happen in tumors in people. And so now we're back in the lab trying to find ways to interrupt this process and to do it in a way that will be most beneficial to people.
Hello, Stephanie.
Hi.
How's it going?
Good, how are you?
Some of the contributions and support that we got from the Phoenix Friends very early was put us onto this track. So right now we know that we have antibodies which we can humanize, and that's our big target right now, is to make those antibodies that we can use them in humans, and then that can be used to sort of vaccinate people against these tumor cells that are going to move to different parts of the body before they get there. One of the unfortunate realities of basic research is that often when you try to get support for your work, you almost have to have all the work done before you can get that support, that next level of support. So, what we would call a pilot project are some of our best ideas that we really have no data on. We have no compelling evidence to show a national agency. So in this case, we knew that these peptide leads were very important and very good, but we couldn't show it. We didn't have the support to do that. And the Phoenix Friends stepped up and gave us some support to do that. And I'm very happy to say that now these compounds have been gone to third and fourth generation. They are active in myeloma. They're not active in the cancers we were how we discovered them, but they are very active in myeloma, and that's being studied at another cancer center.
I think of it as finally getting a liftoff for some of these projects, and now we're finally flying. Last year they made a generous donation to our research program, and then helped us to acquire some data that we put into a grant proposal for the NIH, and that's resulted in a $2 million grant that the NIH is now funding for our research. I use that same type of data in another grant proposal that they just very recently funded for another $3 million. So we've been able to bring in about $5 million.
Well, I just want to give a heartfelt thank you to the Phoenix Friends for all their activities, their support. It's not just monetary support, but their emotional and moral support. When they come and visit us, it's a big shot in the arm. And when we go see them, we love their enthusiasm, and we love the fact that they're there for us. And we feel that they're there for us. And in knowing somebody's got your back like that is really, really important.
Yes, well, I really can't thank the Phoenix Friends enough for their generous support, not just financially, but emotionally too. It's like I mentioned about the researchers here, it takes a village to perform this research. Well, it takes a village even beyond these walls, a community who's really focused on cancer research and cancer care. And it's been a tremendous benefit for our own personal research, our collaborations with others here on the Cancer Center, and I really am excited to continue working with the Phoenix Friends in the future.
There aren't really words to express the gratitude that we have for the Phoenix Friends of the Arizona Cancer Center. Their membership, both associates and active, have made such a difference in what we are able to do. Together, over the last 26 years, they've raised over $5.5 million. Every one of those dollars has impacted all of our lives. Because of them, we're saving lives.